সোমাকে শোনা যাচ্ছে না।
হ্যাঁ, সোমা তোমার মাইক। অ্যাকচুয়ালি আমরা একই ঘরে আছি বলে বন্ধ রেখেছি।
এখন বন্ধ রেখেছি, নাহলে ইকো হয়।
হ্যাঁ স্যার, স্টার্ট করছি তাহলে, 11টা বাজলো। Welcome once again in Bioingene Plant Science webinar series. It's our honor today. We have a very eminent scientist, experienced scientist from Dhaka University, Bangladesh, Dr. Professor Rakha Hari Sarker. Sir, working on plant tissue culture and others, it's from tissue culture to genetic modifications of plants. It's a long journey. It's a long experience, and we're glad we have some chance to share, to learn something from Sir's webinar. I request all the participants keep patience. At the end of this webinar, we share the feedback form submission password. Don't rush for that. We will give multiple time in this YouTube chat box. Please put your questions here. We will collect the questions and ask to Sir after his presentation. I request Shoma to introduce Bioingene and introduce our guest speaker. Shoma.
Welcome to the Bioingene webinar series. The webinar series is a platform from which plant scientists can present their research to the world, and future scientists can gain knowledge, perspective, and inspiration. We welcome all the viewers who have joined us from around the world. If you are new to Bioingene, please note that after attending today's talk, you can apply for a certificate of participation. For this, you need to submit the feedback form that you have received in your email. The password will be provided multiple times in the YouTube chat after the presentation. When you fill out the feedback form, please mention the full name of your institute and its full address. You can collect your participation certificates after 2 days from our website. Bioingene does not send certificates through email. Please visit our website to register for upcoming webinars. You may submit articles in the Bioingene Plant Science Journal, that is a peer-reviewed open access international online journal. We seek to publish papers that are relevant in the fields of plant science research. Selection of manuscripts for publication will be based on novel research, original data, synthesis, or concepts without plagiarism and violation of any copyright. We welcome submissions from all fields of plant sciences, including agriculture, and from all sources. Please visit our website journal section to know more. You may also send your published posters to be displayed on our website. Please contact Bioingene and connect with it via Facebook, YouTube, Twitter, and LinkedIn. Today's webinar is on the topic Plant Tissue Culture, Micropropagation to Genetic Transformation by Dr. Rakha Hari Sarker. Professor Dr. Rakha Hari Sarker has been involved in teaching and research at the Department of Botany, University of Dhaka since 1982. He has obtained his M.Sc from the Department of Botany, University of Rajshahi and Ph.D. from the University of Reading, UK. He was awarded a Commonwealth Scholarship for his PhD. Apart from his PhD program, he has received various postdoctoral and advanced training in plant breeding and biotechnology, plant genetic engineering and biosafety from Germany, CIMMYT, Mexico, International Center for Genetic Engineering and Biotechnology in New Delhi, India, and the Cornel University, USA. Professor Sarkar has received the Bangabandhu National Agriculture Award, the highest state award given by the government of Bangladesh in 2018 for his contribution on research in agricultural sciences. His field of specialization is plant breeding and biotechnology. He has a variety of research interests for the improvement of crop plants using modern breeding techniques and biotechnology. He is also interested in propagation and characterization of medicinal plants as well as indigenous plants of economic and environmental importance. He has published more than 100 research articles in several national and international journals. He is co-author of 4 books published by international publishers. He authored the book called Wealth of Bangladesh Medicinal Plants, published by National Museum of Science and Technology, Bangladesh. He has so far supervised more than 50 students in the field of plant breeding, tissue culture, and plant biotechnology. 8 PhD research fellows and 2 MPhil students have obtained degrees under his supervision. He has also supervised 1 postdoctoral fellow. He is serving as executive editor of Plant Tissue Culture and Biotechnology Journal and one of the editors of Bangladesh Journal of Botany and Bangladesh Journal of Scientific Research. He is the director of Dhaka University Center for Advanced Studies and Research in Biological Sciences. He is also the chairperson of Institutional Biosafety Committee of University of Dhaka. He is a scientist member of the National Executive Committee on Biotechnology of Government of Bangladesh and member of the core committee of the National Technical Committee of Crop Biotechnology of Government of Bangladesh. He has received research grants from US Department of Agriculture, Bangladesh Academy of Science, Ministry of Education, Science and Technology, Government of Bangladesh. He has participated in several international conferences and seminars and has organized several international conferences on botany, plant tissue culture and biotechnology in Dhaka. He is the national coach of Bangladesh Biology Olympiad and jury member of International Biology Olympiad. We are highly honored to have him here on the Bioingene platform. I would now request Sir to please share his screen and begin his talk.
Thank you, Shoma, for a nice introduction. And I'm so grateful to you. First of all, I would like to welcome all the participants around the world. And also I would like to thank Bioingene for arranging this webinar. And particularly, I am personally thankful to Dr. Shubhabrata Mujumdar, the founder of the Bioingene, for inviting me for this seminar. So I think all introduction has been made. So, the area of my research and research activities, I will try to focus on this. And particularly, I think the younger students and colleagues and researchers would be very interested to know what is actually going on in the Department of Botany, Dhaka University, in the biotechnology and breeding areas. So I think I straight away I can go to my presentation and see, I can start straight away. Just a moment. Yes. Okay, I think you can all see my slide.
Yes, sir.
Okay, good. So you see, the idea is the presentation is actually the topic of the presentation, like design, plant tissue culture, micropropagation to genetic transformation. So this is the slide actually provided by Bioingene. So just for your introduction. So next step. So you see, the plant tissue culture and the micropropagation and the genetic transformation are actually very much related topics. So I have designed something like that. I will be talking about the tissue culture technology, applications and its advantages. I will be talking about the micropropagation, then application of the micropropagations, and then I will be talking on plant genetic transformation, and at the same time the role of tissue culture for transformation, and also the various issues related to the transformation. So let's start with the presentation. The plant tissue culture is a good old technology, and for plant tissue culture, actually, we need to maintain and grow the cells and tissue or organ under sterile condition. So that is very important. Then you have to maintain a sterile condition, and you have to choose the right plant material, right explant for your culture. So tissue culture is widely used to produce clones of a plant in a method known as the micropropagation. So we'll be talking about the micropropagation as well. So, so this is, in the beginning, I would like to mention some of the things that really, very elementary ideas. So what is actually the initiative for the plant tissue culture? The totipotency is the main issue for the plant tissue culture. So totipotency is the ability of the cell or tissue or organ to grow and develop into a fully differentiated organism. So with the help of a little tissue, you can develop a new plant. So all cells have the potential to develop into a new plant. That is the main idea behind tissue culture. So this is the big history of tissue culture. I'm not going to discuss all of them. So you see, from 1902 to here we have 1950s and 60s. I would like to mention here the botanists actually has a huge role to turn the idea into an effective one. In the case of the plant development, in case of the practical application, the tissue culture, the botanists actually take the initiative for this. So before that, there are a lot of huge ideas for the tissue culture. People working on the tobaccos, people working on the various other materials, and see how the plants actually respond to the cultures and so on. So then in the next slide, you see from 1960s to recent time, we have a lot of different activities, like Professor Cocking from Nottingham University, who developed isolation of the protoplast, which is very significant in the research of the tissue culture. And at the same time, 1962, Murashige and Skoog developed MS medium. We all know that what is the MS medium, that this is the basic basal medium for tissue culture activity. So we use the Murashige and Skoog medium in many tissue culture activities. So Murashige and Skoog in 1962, they have developed this tissue culture medium. Then there's another interesting thing happened in 1966 when Shipra Guha Mukherjee and Professor Maheshwari in Delhi University, they developed the haploid culture. So haploid culture is actually very important for the plant breeding, particularly, anther cells can be converted into plants, and then you can produce the haploids and you can use them for breeding purposes very nicely. And, there's a lot of breeding actually been done with the help of the anther culture, particularly in the rice breeding. So then other things, gradually the molecular biology actually developing. So all the activities regarding the biotechnology is actually crystallized that time. So the discovery of the restriction enzymes, restriction endonucleases particularly. Then also in the 1972-73, Cohen actually developed the recombinant molecules. So this is also very important. And then in 1974, actually the development of the TI plasmid in Agrobacterium tumescens, which is also very important. So we all use Agrobacterium for transformation. So it is very important that, Agrobacterium mediated transformation, that technology actually developed from that time. Then, the role of the TI plasmids also been discovered at the same time. And then in 1983, the first transgenic plant was actually developed by Monsanto. And so we all know the Monsanto is a big company in the field of biotechnology. So they developed the first transgenic plants, and simultaneously there are a lot of work going following the transgenic. So I'm not going to discuss after that, because we all know the after 1985 there's a lot of activities going on in the plant transformation area. So I'll be going to some other aspects on the tissue culture gradually and transformation. So you see, for the concept, how the tissue culture actually work. So this is an elementary slide, elementary idea regarding how the plant actually de-differentiate, how the plant actually regenerated. So you can take some part of plant, particularly have the seeds actually been shown. So you can get the embryos, and then you have a hormone mediated medium. So you have auxins, and then you can develop callus, and then you can have a combination of auxins and cytokinins that will make the callus into a plant. So you can convert the callus into a plant, which is actually main theme. And the balancing of the hormone, growth hormones particularly, is actually very important for the conversion of the callus tissue or any kind of tissue to a complete plantlet. So this is very important for the tissue culture as well. So you see, I'm going to show you a couple of other slides regarding the tissue culture activities. So explant is very important. You have to choose the right explants, and the living condition of the tissue. What is the status of the tissue is very important. Leaf tissue you can take, you can root, zygotic embryo, and whatnot. You can take many different kinds of explants for your culture. And also very important for the tissue culture is to have a mineral nutrients. So we have lots of mineral nutrients requirements, like the micronutrients and the macronutrients as well. So you have to choose the minerals, and then the carbon source is very important. So we normally use sucrose, and sugar can be used, so it's no problem. So you can modify the media according to your requirements. And of course, the plant growth regulator, which is very important. So you have to choose the right growth regulators for a particular plant material, and you have to choose which explant actually you're talking about, what type of plant actually you're talking about. You're taking, you may be, some kind of plants which are very responsive to tissue culture. Some other plants is not been very responsive to tissue culture, like the tree plants, which are, normally the trees are very difficult to regenerate. So you have to choose the right type of hormones as well. Then see, we have callus. The callus is a mass of parenchymatous cells. So when you actually put the things into your media cell, these photographs actually show you some little bit of callus here. So you can develop callus, and from the callus you can actually develop either organ, particularly you can develop a shoot like this. In this photograph you can develop a shoot, and also you can develop embryos. So I'll be going to show you some of the pictures of the embryo later. So you can develop embryo. So it is somatic embryo. So normally plant, after sexual reproduction, they produces the embryo, which is actually a zygotic embryo. But in the tissue culture, you can actually develop somatic embryo. So the embryo, the nature of the embryo is that it will be developed into a complete plantlet after putting them into the right medium. So these are the basic ideas and the basic requirements for the tissue culture. Then organogenesis. I'll be talking a little about regarding organogenesis. Organogenesis, I have already mentioned that you have to develop organ. And particularly, you cannot develop root in the beginning. You have to develop, for a complete plant, that you have to develop a shoot. So you have to balance your hormone in such a way that you have to develop, your explant could be converted into a callus, and then it is a meristemoid, which it will be developing some meristems, which is actually is very initial stage of the formation of the shoots. Then the organ primordia. So afterwards you can develop a new shoot. So you have to balance the hormones here, and also with the help of the tissue, you can develop with this callus tissue. You can actually convert whatever the things you actually want. So explants, then de-differentiation, then induction of the shoots, and then organs, and then afterwards you have to develop the roots for developing a complete plantlet. Without the root, you cannot actually cannot get out from the medium, and you cannot acclimatize them, you cannot establish them in the soil. So you have to have a root system developed, and sometimes the root development of the root may be a little bit difficult in many plants. So now I'm talking about little about the embryogenesis. So you have I have already mentioned that you have to develop your somatic embryos. So for the somatic embryos, you have to balance your hormonal requirements. So normally we have explant, then we could use 2,4-D, which is actually a kind of auxin, which actually develop lots of callus, and then you have to convert the callus into an embryogenic callus. Then you can benzene adenine or zeatin, whatever the requirements is necessary. You can convert the callus into the somatic embryo, and then you can develop into a plant depending on your plant growth regulators. So in case of the dicot, there are two huge work actually been done in the area of the embryogenesis. So there are people use different kinds of media, different kinds of different kinds of sources of the material. So for the development of the embryos. So here, in case of the dicot, you have the globular, heart-shaped, torpedo type of embryo, and the cotyledonary stage you can eventually get, so you can develop into embryo. And then monocot, you have a globular and scutellar tissue or the coleopterar tissue. You can use them for the conversion of the embryos in case of monocots. So, and at the same time, there are a lot of activities actually been done in India and some parts of the world, particularly in India. They have developed synthetic seeds out of the embryos. So you can develop, the embryo and you can encapsulate, and then whenever it is required, you can convert, you can develop into a plant. So this is it's a very good technology for the tissue culture. So at the same time, there's another technology which is very important, and also it is one of the greatest scientific achievements regarding the finding out the protoplast and isolation of the protoplast and fusion of the protoplast. So it is a somatic fusion you can do. So we can develop the somatic hybrids. We call it the hybrids we can develop. So in this case, we have to find out how to develop the protoplast, how to isolate protoplast. So I have already mentioned that in 1960s, Professor E.C. Cocking of the Nottingham University and his team, find out the isolation of the protoplast mechanisms, and they develop the protoplast. So in case of the protoplast, you don't have to have a cell wall. So you have to eliminate the cell wall and get the protoplast out, and then you can develop into whenever it is requirement. You can fuse them, and you can develop into somatic hybrids, and then you can develop into plants. But you see, it's the technology is fine, but the problem is, most of the cases actually it is very difficult to generate plants from this kind of protoplast. So people actually develop, and during recent days, people have been more enthusiastic regarding the protoplast, because some of the new technology is actually coming up. So probably we have to try more on the protoplast isolation mechanisms, and also to find out how to use the protoplast for practically for the biotechnology purposes. Also in the CRISPR technology, protoplast is also very important. So this is one of the areas that we will be interested in the future. So now this is another one, which is actually haploid culture. I have already mentioned that Professor Shipra Guha Mukherjee of Delhi University in 1964, they have developed this. They develop is kind of, the story is that they develop very casually. What happened? They actually Shipra Guha Mukherjee and Professor Maheshwari actually was involved in the Datura anther culture. So develop anther culture for Daturas, and there's lots of tubes there. And some of the tubes actually they said it's not been responding for a long time. So they keep them outside of the lab. So in the corridor it was there, and suddenly they found that in this culture, some green spots are coming up from the anthers. Then they eventually they find out that these anthers actually developing, the cells and the tissues, and also, eventually they develop into a plant. So this is the history of the development of the anther culture and the development of the haploid plants. So then it was their invention. They find out how to develop an anther-derived plants. And also they develop, afterwards they develop microspore. What is the importance of the haploid culture? Because if you have a haploid plants, you can develop dihaploids. So dihaploids is very important for the plant breeding, because if you cross two plants and you have a F1 plant, from the F1 plant, actually, if you produce the anther culture, anther-derived plants, so you have a set of plant with a haploid set. And then you have another, plant which is actually you want to develop into a diploid. So you can develop from the haploid. You can eventually develop a diploid plant, which will be more homozygous in nature. So it will be, it's very good for the plant tissue for development of the, homozygous plants through tissue culture, anther culture, and also for the breeding. So this is very important for the development of the haploid plants. And tissue culture is actually the main, without the tissue culture, you cannot do this haploid production. So this is the one idea that you can use in the future. Now, there are several applications of the tissue culture. I'm not going to discuss them for all in details, but you see, their haploid production I have already mentioned. So rice, wheat, barley, and then also the triplet production, which is actually used for the fruit trees, fruit plants, embryo rescue. This is very important for the wide hybridization. There are many cases we actually make interspecific crosses, but in that cases, actually, it is very difficult to maintain the zygotic embryo eventually, because some incompatibility and some barriers are there. So you can rescue the embryos to a culture, tissue culture media, and then you can generate a plant. So this is also very important, embryo rescue. Then the somatic hybridization, which is actually have been mentioned, that the somatic hybridization can be done with the help of the protoplast culture, which is actually a good avenue to work. And then somaclonal variations. Somaclonal variations is very important in the sense that in the tissue culture media, you can actually create some variations. So those variants are actually very important. In some cases, actually, particularly for the plant breeding, we have problem that we cannot get lots of variations. So somaclonal variation could be one of the opportunities, one of the options that actually you can develop somaclones in the media, and you can use them. Recently in our lab, we have been able to develop some somaclones from the brassica cultures, which is very interesting, and we'll be looking into it in the future. So sometimes people thought that somaclonal variation is not very effective, but you see, there are lots of reports are coming up regarding somaclonal variants in Bangladesh. Sugar Crop Research Institute, they have also developed very nice somaclones for high sugar content, and also some to some extent they are actually disease resistant, and some kind of achievement they have developed. So somaclonal variants is still is very good. Then you can produce the disease-free plants, which is very important. Particularly the plants like vegetatively propagated plants like the potatoes, like banana. So you can produce with the help of the tissue culture, particularly meristem culture, you can develop disease resistance plants. So everybody actually know the technology. You have to take little bit of the meristem and then you can develop into a plant, because you see meristem is important since there is no vascular tissue. So there is no transmission of the virus within the vascular tissue, and so the meristem is actually free from virus. So you can develop virus-free planting material for plantation, and also virus is a big problem for potato and the banana as well. Maybe some other plants, you can use them for disease-free planting material. Then clonal propagation. Clonal propagation is actually a part of the micropropagation activities. So you can huge area, and this is the area that actually commercially been used for many companies all over the world, particularly India and the Netherlands, Thailand, Singapore, Vietnam. They use the clonal propagation very nicely and efficiently, and these are actually they are in a business of commercializing of the tissue culture technology, and many industries actually develop. Even in the Nepal, they have also the micropropagation industries growing, and of course India is a big, and there are big companies in India as well. And also the people actually working on the cut flower business, they like the orchids and some ornamental plants like Jarbera and tulips. They use this kind of clonal propagation very efficiently and nicely. Then number 8 is actually is another important thing is the second production of the secondary metabolites. This is very important for development of the metabolites, finding out the metabolites from the tissue culture using the tissue culture technology. So you can get lots of products, metabolites, particularly for the use for medical purposes, for the cosmetic purposes. They can be extracted from the tissue. So you don't have to, in these cases, you don't have to develop into a plant. What you have to do, you have to raise the tissue. You can do the in vitro culture without organogenesis. Only the callus tissue, you can develop huge number of callus tissue. You can generate within a bioreactor, and then from the bioreactors you can actually extract the secondary metabolites and can be used for very specific purposes whenever it is required. And then the number 9, which is actually gold technology, for the conservation of the germplasm. And the cryopreservation particularly is very important because in the low temperature you can preserve the material for a longer period. So you have a technology that you can preserve the material for a longer period, and also not only the cryopreservation, but you can also preserve the germplasm, even in the normal tissue culture for a longer period, and so you can actually preserve, you can actually conserve very important germplasm. So these are the various aspects of the tissue culture and applications. So I'm going to the next slide. So in brief, so micropropagation is one of the priority, germplasm preservation, somacronal variations, and embryo rescue, haploid production, and also the protoplast fusion. These are the messages that actually we can use from the tissue culture. So this is tissue culture is, although people used to say that already tissue culture has been exploited, but even though several activities being going on, and in the future probably we have to continue with the tissue culture for our specific purposes, and we'll be using the benefits of the tissue culture in the future. So now I will be going a little bit about the micropropagation. So here micropropagation techniques actually been mentioned that in the micropropagation, so you have the possibilities that you can rapidly generate lots of plants rapidly in a small environment, in a tissue culture growth room, and thousands and millions of plants can be propagated. And in this case, actually, you have to find out, the specific explant, of course, and you can actually, without going outside, you don't have to collect the explant every time. Whenever the material that you have in the beginning, you can use them for repeated culture. So you can produce this through subcultures. You can produce millions of thousands of plants, and those are actually being used for the commercial purposes. So I will be giving you some of the ideas regarding this. So micropropagation is actually it's a tissue culture based technology, of course, and it's sometimes it is mostly used for the asexual production. So it is a clonal propagation of the plants. So you can use them for your according to your requirements, and it may be some of the ideas that confused with the tissue culture. Actually, micropropagation refers to the in vitro multiplication and regeneration of the plant material under aseptic conditions and under the controlled environment. This is very important. So, and whenever actually required. So you have to develop your own environment conditions so you can have a large culture maintenance, and you can do the micropropagation with so many advantages. So here you see, this is somebody said that this is the art and science of multiplying plant in vitro. So micropropagation is actually in some of the labs, particularly sometimes it may be a bit difficult that maintaining the environment, particularly avoiding the contamination. Contamination may be a problem, and the fungal contamination or bacterial contamination. So culture contamination is a problem. So we have to handle that properly. So in brief, so you can choose different kinds of explants. I would say leaf tissue or the shoot tip, and also axillary buds, nodal segments, root segments. You can use them for micropropagation. So, and then you have a axillary buds as well. So you can develop huge plants, in vitro raised plantlets, particularly shoot you can develop, and in that case you have to develop also root for the development of the complete plantlets. So you see, this is one of the plants that actually we've been working on, Vitex trifolia. This is a medicinal plants, and the Vitex is actually very important for medicinal purposes. So you see, there's very straightforward propagation. So you have a nodal segment, and you can raise huge plants, and then you can have a good root system you can develop. So this is the system that you can do. Then this is another, this is from our lab. This is the propagation of the Gerbera plants. So you see, the Gerbera plants can be developed from tissue culture from the inflorescence. So you can have a flower bud. So from the flower bud, you can choose the explants. You can choose lots of different explants, but flower bud is the most responsive in case of the Gerbera. So you can have the development of the shoots from the Gerbera. So you see, this is the straightforward micropropagation, and then you can have a rooting, and you can do this. And also these are the plants actually been transferred to field for flowering. So this is a nice technology, and Gerbera is very important for many parts of the world. Even in Bangladesh, actually, there is no use of the Gerbera in the 4 or 5 years back. People didn't know anything about Gerbera, but there is, if you go to the market, there are lots of different kinds of Gerbera is available here, and all been cultivated in Bangladesh. So you see, you can have a hormonal balance for the micropropagated plants, and you can have convert with the help of hormonal balance. You can convert the plant into the development of the root system and development of the huge subcultures to generate lots of plants. You can generate with the help with the balancing the hormonal, particularly auxins and cytokinins. So I'm now, so this is the picture for the shoot organogenesis. I have already mentioned that regarding the shoot organogenesis. So this is the slide. Then you have this slide actually showing you the somatic embryogenesis. So I have already mentioned that you can convert the plants into an embryo. So you see there are nice embryos developing from the suspension cultures. So these embryos will be germinating into a new plant. So it's no problem. So you can have huge number of plants you can generate through this somatic embryogenesis. So then you can develop a bioreactor, so you can automatically you can control the whole propagation system. And many European countries, they actually do it. And of course, in the Netherlands, or also some other countries like also in USA, and they use the bioreactor for the maintenance of the media and the culture media. They can transfer these, and they can have huge number of plants, which is actually automated. You can do this. Particularly in Netherlands is very good in this technology. So they have actually invented lots of bioreactors and the production system for micropropagation system in using the automated system. Now, here is a problem. You see, if you work with lots of different materials, one of the materials that we've been trying is it's momi plant. is very rare in, I think in all parts of the world, it's very rare species. The plant is actually, we are actually trying to develop, regeneration system for this plant. You see, the plant is actually not responding very well. And you see, when the plant has a latex, and also it's a very quickly became, change due to, lots of phenolics actually generated, so you cannot maintain the culture. So it's a tree species. You see, this slide actually have given, so you can encounter with lots of difficulties during this culture system. So you have to modify the media, and you have to, and probably it will be very difficult to regenerate. And in this case, actually, what happened to us, we found some seed, and through the seed culture, we've been able to generate plants. So this is one of the difficulties regarding micropropagation as well. So now I think I have completed the tissue culture part of my discussions. Now I'm going to talk about the transformation and why transformation is actually required, plant genetic transformation, and we'll be talking about the various issues relating to the transformation, particularly in the Department of Botany, what we're actually doing for the transformation. We'll be highlighting on this, our research activities. So let's go to the next slide. You see, why the transformation is actually required. A lot of questions regarding whether we shall continue with the transformation, whether we shall be going for the genetic modification of the plants, and what is actually background behind it. You see, this is the slide. You can see that how the arable land is actually decreasing. So in 1960, the per head arable land was 0.44 hectare per head. In 2000, it is actually changed to 0.22. So you see, in 40 years, it has been changed to reduced to 50% in this case. And then in 2050, the per capita per head arable land will be 0.13. So you see, the arable land is actually decreasing, but the population is not decreasing at all. So you have to feed the people. So you have to develop new crops. You have to develop and modify the crops, and you have to maintain the yield potentials and the yields of the plant. So this is one of the slides that actually very important to understand why transformation is actually required. So this is the story about the Bangladesh. So in 1972, we have 75 million people. And now we have more than 170 million people. So huge people actually, huge population we have. So in this case, actually, we have to increase the productivity, and also some other areas, not only the rice, because we are a rice growing country, and people have, without rice, they don't think about anything. So we have other crops like pulses, oil seeds, edible oils, vegetable seeds. So these are actually maize seeds. These are actually our requirements. So you can understand that feeding a huge population is very important to have, and also the maintenance of the nutrition. So this is also very good thing to understand how we can actually cope with the situation. So other problem is that I think not only in Bangladesh, but the problem is global problem like the salinity, flood in cases, and also the drought, and also the submergence. So these are the we call it abiotic stresses. So these are the major abiotic stresses for the crops. So you have to address these things, and you have to develop new plants to cope with this and the conditions like this. And also the other thing that we have the diseases in the various kinds of diseases in plants. So in that case, fungal diseases, also of course the insects are there, and then the bacterial diseases, viral diseases. There's huge diseases, and to get the proper yield, you have to maintain the the health of the plant. So therefore, you have to develop, you have to generate new plant for the disease resistance for this not diseases. You see, also this is very important that people used to spray huge amount of insecticides or fungicides, and these are actually not good for the environment, not good for the human health. So you have to design the crop which is actually good for the future, which is not harmful to the environment. So this is also very important. You can do with the help of the modification of the plants, genetic modification of the plants. So these are also very important issues. Now also the conditions like the flash floods, also some environmental calamities, and this is due to the the conditions due to climate change. We rather you can say that this is the effect of the climate change, and you have to address these problems as well. And in Bangladesh or India and Sri Lanka or Philippines, whatever, everywhere actually we have this kind of problem. So this is the new issues that we have to address. And also the another thing is actually regarding the water. This is the next slide, which is actually water. Although we have lots of water, but we have also crisis of the water. So you see that the water sometimes, without the proper irrigations cannot be done, because all the modern varieties actually require lots of water, particularly rice and other related crops. So you have to develop, plants which can actually withstand less water. And also you see there is an example here. So for the production of the 1 kg of rice, you spend 4,000 to 5,000 liters of water. You see enormous amount of water has actually been used. So in this case, actually, so you have to maintain the economy of the water economy. You have to design your crop. Otherwise, our water will be a big problem for us, and we will be losing all our groundwater, because particularly we use groundwater for the irrigation. And now this is a slide just to give you some idea. You see, this is a slum in Dhaka, where you can see lots of, these are people actually, it's very close to one of the, very popular area in Dhaka, which is, you see, this slum has lots of people actually living there. And you see, they are low income people, and they are actually suffering from malnutrition. And they cannot have, proper nutrition for every day. And they might be, they're hardworking people. They can have, they can actually eat somehow or other, but nutrition is the big problem for them. And also, this is the slide actually we've been discussing regarding the vitamin A deficiency in Bangladesh, in many developing countries. So vitamin A deficiency is a big problem for the nutrition in case of the nutrition area. So vitamin A deficiency is a big problem for us. As you see, the blindness, and not only the blindness, but also some other diseases actually develop, and also the mental development is very poor if the vitamin deficiency is there. So these are the areas actually good to work to develop new crops which actually cope with the situations like the diseases of the plants, the abiotic stresses, and also the nutritional enhancement. So these are the areas actually we can work. So you see, this slide actually I have given here. So the idea of the crop improvement and the crop development and the domestication of the crop is not a new idea. So people from the prehistoric time, people has been using different technologies and the methods to improve their crops. And also, according to the requirements, because by knowing or unknowingly, they have done it in the past, and still, the people are actually searching for, new crop varieties, and for these purposes, people actually travel from one part of the, world to the another area, to the different areas. They're actually moving around to find out, new crop varieties, and which is actually good for, cultivation and the production. So this is the idea that all the time actually people have been using technologies for the for enhancing the crops. Now, also you have the breeding material. The breeding material is actually used for the generation of the new products. So you have the genetic resources, you can use them. And also this slide actually I've been shown here because how long it will actually take for the conventional breeding. So so 10,000 years actually been spent for the development of a maize, modern maize variety from this wild Q70. So you see the huge time is actually being consumed. So in this case, actually, it's a traditional crop breeding is really time consuming. That is, this is the thing that I want to mention here. Also, you have to find out, germplasm. So this is the slide for this tomatoes. You can use them for the wild germplasms, but it will take a huge lot of time to the back crossing and other things actually is a requirement. And also the white crossing. We've been trying in Bangladesh actually the wild rices and the modern rices to hybridize them, but it is still a big problem for getting proper hybrids or heterozygous plants from this and transfer of the genes from wild to cultivated one. So this slide actually I given to remind you that this is the statement from the Charles Darwin. He has mentioned that it is not the strongest species that survives, not the most intelligent, but the ones most responsive to the change. So you have to, he is an evolutionist. So he knows, he's a naturalist, and so he's a famous biologist, everybody knows that. So he has this statement, you can remember that one. So you have to develop your material according to your requirements, according to the change of the environment and particularly the conditions. So so the technology actually been marching through. So 1860, when Mendel actually discovered, genetics, and then discovery of the hybrids in the 1920s, 1950s, then the mutations developed, the 1960s, the tissue culture technology and embryo rescue techniques, 1980s, the transformation technologies and the GMO technologies, and the technology is going on like the CRISPR technology is now developing. So there is continuous processes being going on for the development of the new crop varieties and the modification of the crops. So these are the people. Say I can mention here the two people here, actually Dr. Norman Borlaug and Professor Swaminathan. So they are the pioneer of the Green Revolution. So Professor Swaminathan actually mentioned that we have done a Green Revolution, but we want to have an evergreen revolution. So evergreen revolution is important for us for the future. Now, this is this technology. So we are actually emphasizing, giving a lot of emphasis on the biotechnology. So biotechnology is the technology for this century and maybe in the next century as well. So this technology actually been started with this event of the development of the DNA molecules and the structure of the DNA molecules, which actually is important for all kinds of biotech activities. So the knowledge of the DNA is very important for the biotechnological purposes. So this is discovery of the DNA molecules, Watson and Crick model. And then also I would like to mention some of the slides like the Paul Berg actually developed, the recombinant DNA technology, which is also very important for development of the genetic transformation system and so on. So now biotechnology is of course the potential options for increase the agriculture productivity, and not only in Bangladesh but everywhere where actually is required. And then I'm not going to skip this one. So then I'm just talking about a little bit about the transformation. So in case of the transformation, you have to develop your own construct, and particularly when actually using Agrobacterium. So we can modify the Agrobacterium, and Agrobacterium has the capability to transfer the genes into the plants. So Agrobacterium efficiency. I'm going to the next slide. So show you. So of course the tissue culture technology is actually required for the development of the transformation system. So this is the Agrobacterium technology, where you have the Agrobacterium is actually natural genetic engineer. They can convert, they can transfer the gene into plant from bacteria to the plant system. So this is very important for the transformation. So you have the Agrobacterium, they produce the crown gall. So crown gall is actually the disease, but the people actually what they've done, they have actually modified the Agrobacterium plasmid, where actually they call it disarm TI plasmid. So disarm TI plasmid is actually used for transformation. So, and you see, you see this picture. So how big is the tumor is actually developed from due to the Agrobacterium. So people actually use, the scientists actually use this technology for the transfer of the genes. Agrobacterium is not harmful to the plant, but only they produce crown gall. And this, the crown gall technology has actually been used for the transfer of the genes to your plant, particularly specific plant. So you have, this is the slide to show you how the Agrobacterium actually transfer the gene. This is the mechanism. So I'm going to describe it in details. Now, this is the slide. You can get an idea that you have Agrobacterium, and you can generate the Agrobacterium culture. You have a plant material, plant culture, particularly the callus you can use, or any explant. Then you have to infect this explant, and from the infection you can get a plant. Also, you can also use the in plant system as well. So this is the way actually transformation being done. Also, the Genegun can be used for the transformation, which is actually not an Agrobacterium mediated system. It is the directly, the system is actually used for a biologistic system. So you can directly insert the gene into your plant tissue. So this is the slide that everybody knows, that how the tDNA is actually been changed to genomic DNA. So this is the transfer of the tDNA into the genomic DNA of the plant, your requirements. So this is the, you can actually customize the tDNA, and you can have your own construct, and you can develop this for your purposes. So this is the slide. Actually, everybody knows that the traditional system of plant breeding, you can transfer the genes, but at the same time you can transfer the other genes as well, other traits as well. But in case of biotechnology, you can directly transfer the specific gene into your desired plant, so, and at the same time you have a desired gene. So this is the difference between these two systems as well, particularly. Now I am going to talk about I think we have quite a lot of discussion, and then I'm now going to discuss how the time is. This time is actually limiting. Anyway, so I'm going to discuss some of the activities of the biotechnology and the genetic transformation work in the Department of Botany, Dhaka University. We are working on lots of grain legumes like the lentil, chickpea, peanut, and mung bean. And also, we also work with the brinjals, potatoes, the brassica, tomato, and rice as well. So, we have huge, activities, and we have a good team to work together for different crops in our department. And you see, these are the problems of the lentil. We have diseases and also the insect infestation. So we have developed the transformation protocol for lentil, and also chickpea. We have lots of problems with the fungal diseases, ascochyda blight. And also, for peanut, we have also the problems with the, the ticca diseases, which is actually spot due to the fungus. So lip spots are very, prominent in peanut. So we address this crop as well. And also the virus in case of mung, which is actually yellow mosaic virus, is a devastating disease for mung bean. And so you see, breeding in case of these crops, actually breeding is not been possible because of the they have a sexual incompatibility, they have a narrow genetic base, and particularly not available and not adequate genetic resources actually available. So we cannot actually do the conventional breeding. Therefore, it is important to have a transformation for these plants. So we use the Agrobacterium mediated transformation for these purposes. And so you develop a protocol for the development of the regeneration system, and at the same time you develop a protocol for the transformation system. So we use in lentil, we use lots of different kinds of explant, and so these are the cotyledon attached embryos. We use them particularly. This is very good for lentil, and you see these are the different explants used for lentil transformation and regeneration, and these are the cultures for the lentil regeneration system. You see, these legumes are actually very recalcitrant. They do not respond very spontaneously. They have a problem in the regeneration. Everybody knows that legumes, not only the grain legumes, but also the leguminous plants, big trees and things like that. They have also the same problem. And also we have a problem with the rooting. So rooting is a big problem for lentil particularly, we have found. So although, there are some other techniques actually developing to overcome this problem, but at the same time, we have developed a technique which actually bypass the in vitro system of rooting. We develop a flower under in vitro conditions. So we develop, in case of the lentil, we develop in vitro flowering system. And so you see that you can develop flower in the in vitro condition. So if you have a transform plant, shoot even, you can develop flowers. And these flowers can be, they produce seeds under in vitro condition. And these seeds are actually transgenic seeds, you can use them. And so these are the gene construct. I'm not going to discuss about this. So we have a specific purposes for the gene construct. You have to have, for all transformation, you need to have a, construct for your purpose. And also you have most of the cases actually we assay these transforms with the help of the GAS genes. So these are the GAS expression of the lentil explants, and also the whole plant actually showing the GAS expression, and also the seeds and other part of the pollen grains and the seeds also have the expression. And also another big problem is that when you're actually doing the transformation, you have to develop a system of the selection. So development of the selection of the transform, most of the cases we use antibiotics. So the antibiotic selection is very critical. So you have to find out which dose of the antibiotic, which concentration of the antibiotic is suitable for your explant, for your material. So you see here in this slide, we have these are the dead tissues, this dead plant. Mainly the shoots actually is dying, but some of them are actually surviving. So they are actually surviving under, stringent antibiotic condition, stringent selection. So you can choose them for your selected material for, development of the raising of the transgenics. So this, at the same time, you have to do lots of molecular work, molecular assessment, particularly for the find out whether the gene is there or not, how the gene is actually expressing. So you have to use the molecular techniques like the PCR technology, the RT-PCR technology, then the Southern. You have to do this, so confirm that your plant is actually transgenic. So we have actually published some of our work in vitro, and also we have published in a book by Elsevier. So these are and then I quickly go to the mung bean, because I'm not going to discuss it. The story is almost same for all legumes. So you see, you have to develop your own construct, and in this case, actually, you have to have the idea from the bioinformatics as well. So you have to design your construct, and also your primer designing. You can do this. So this is the idea. And also, development of the construct, which is very important for your. So you see, you can customize your plasmid, and then you can use them for specific purposes. And so these are the cultures for the mung bean, and also the regeneration of the plants. But you see, in some cases, actually, rooting is a big problem. So in that case, actually, you have to do some in vitro grafting even. So we have developed a technology for the mung bean. For the grafting, you see, this is the grafted tissue, grafted plant material here. So you see, if you do the anatomy, you can see how the grafting actually established. So through the grafting, you can develop, new, even you can develop seeds. So these are actually grafted, from the grafted plants, you can develop seeds. So this is one of the options that you can do with the help when the rooting is a problem. And also you can have the GAS expression for particularly to confirm that this is the transgenic is going on. But of course, sometimes it is chimeric in nature, but you have to find out which is actually suitable for you to modify your media, modify your condition, and then you can do this. So at the same time, we maintain the biosafety regulation. So you have to maintain, you don't mixed up with the other plants. You have to isolate them. You have to grow them, differently. And after taking lots of care, so there's no contamination for the plant material, because these are transgenic plants. So you have to maintain that. Primarily, we don't have this kind of facilities, but gradually we have developed biosafety facilities in our department. So we are actually now, we can maintain that, biosafety and other protocols, required protocols there. So these are some of the slides, our students actually maintaining the biosafety issues here, addressing the biosafety issues, following the guidelines. These are the plants actually developed transgenic plants of the peanut. These are actually growing in the greenhouse at this moment. And also potatoes, we have also done the potato culture as well. But the potato is comparatively, potato is much more, I'm not saying it is easier, but it is more, nice to work with the potatoes. And also, so you can get the potato tubers in tissue culture conditions, and also you can get the potato tubers in the normal, conditions as well. So these are the confirmation of these transgenics. So you see huge, nice potatoes have been growing, and these are all transgenic potatoes developed in our lab. So then we are working on the salinity and the drought tolerance. Grateful to Dr. Narendra Tuteja, from whom actually we have got the construct, PDS 45 construct. So we are actually using them. So this is the tissue culture media and the tissue culture conditions developed for the brinjals. So this is the Solana melongena brinjal. So we have a protocol for the development of the raising of the plants, regeneration system, and also you see the plants are actually nicely the flowering and have fruit setting is nice. And then we have a primarily we have tested with the GAS gene, so it is working in the GAS genes. And then we use the PDS genes as well for the transformation. So this is the confirmation of these PDH 45 gene for confirmation of the piece through the PCR technology that the gene has actually been properly transferred. So these are the plants, transgenic plants as well. So you see these are the plants of the nice brinjals are growing. Also, we work on the tomatoes. I'm not going to discuss. I think the time is actually constraints, so I'm going to complete this presentation. So tomatoes, we have also this, we are also using for the tomatoes for selection of some of the summer tomato-like things. Actually, temperature responses to tomatoes we have been working on. So we'll be in the future we'll trying to find out which tomato can survive under drastic environmental conditions and so on. So we are working on this area. So these are some of the molecular work that we are trying to find out, the nature of the expression of the tomatoes in particular condition. So this is the more PCR results. And then I would like to, this is an acknowledgement. But primarily actually we have got lots of help from the Professor Hans Jakobsen from the Hannover University. Also, we are grateful to Dr. Narendra Tuteja, Dr. V.S. Reddy from ICGEB New Delhi for providing us technical support and the gene constructs, and the funding actually been raised by USDA. So we are grateful to the US Department of Agriculture for helping us in this huge activities in our department. So this is the, I would like to also, grateful to our students like Shubhrata Kumar Das, Tanjin Akhtar Banu, Sujoy Kumar Bhajan, Sanjida Rahman, Mullika, and Sabina Yasten. There are some more other actually, as I didn't mention that, but there are some other work, and M.Sc students are also involved in this activities. And also grateful to our professors, Professor Ahmed Samsul Islam, Professor Mozammil Haq, and Professor Imdadul Haq here, Professor Nul Islam, Dr. Rita Saraborna, and Tahamina Islam. And they are my colleagues and present and past colleagues. So I'm grateful to them for their kind support and throughout our journey. And now I will be in 5 to 6 minutes, I will be completing regarding the probiotic activities in Bangladesh, because this is very important. And so you know that the Bangladesh is actually in the GM crops, Bangladesh is very popular now because of this technology. So Bangladesh actually approved the BT brinjal in Bangladesh. So in 2013, it has been approved. 4 varieties of the brinjal been permitted to cultivate in a limited scale. So it is now growing on. So you can see that infestation due to the fruit and shoot borer, and also this is the development of the fruit and shoot borer here. So you can compare these 2 plants here, particularly the 2 fruits. So this is the some of the visits that we've done during the confined field trials of the BT brinjals during early before 2013. So it is actually 2010 and 12 and some of these periods. These are the slides from that time. And also you remember that Bangladesh is actually working on the golden rice. So it is very important for Bangladesh to develop a golden rice, and the Bangladesh Rice Research Institute actually working on the golden rice. So you remember this picture. This is the Professor Engoproticus, who actually inventor of the golden rice technology. And then some of the trials actually been going on in the Bangladesh Rice Research Institute. Some of the visitors actually have been looking into the development of the golden rice plants in the confined field trial as well. So we are hoping that the Bangladesh will soon allow the golden rice to be cultivated here. And recently, you know that the Philippines actually have given the permission for the cultivation of the golden rice. So a lot of discussion is going on at this moment in Bangladesh. So hopefully, we hope that golden rice will be permitted very soon. And at the same time, when you actually do the biotechnology, you have to have a good biosafety system as well. So according to biosafety protocol, Bangladesh is actually signatory of the Cartagena protocol. So according to the Cartagena protocol, you have to develop your own biosafety system, and Bangladesh actually been able to develop the biosafety system. So with the help of the biosafety protocol and the national framework of the biosafety, we call it NBF. So with the help of these two documents, we can work in the laboratory, and also you can apply for the permission for releasing of the crops. So this is the technology, this is the protocol is here in Bangladesh. So and have to have a good evaluation procedure for the biosafety and as well as for the product. And each country should have this guideline, and they have developed their own system to assess the GMOs, and also the environmental issues like the issues of human health and the environment is of course for the important to address the biosafety issues. And also you have to have a good system of the risk assessment and the risk management. Those are the terminologies actually being used for the biosafety people. So you have to understand what are the terminologies and how you can address these issues. So ultimately and eventually you have to go through this protocol, and you have to develop your own props for the we call it GM props, which will be suitable for the genetic engineered crops for the future. So this is the protocol. So we have to go through this. You can apply. You have to have a good assessment of the risk, and then you have to risk elimination process if it is required. And then also you have GMO detection system, and eventually you have the decision making authority to approve whether it is suitable or not. So since 2000, Bangladesh actually been working on it, and recently Bangladesh has developed their own biosafety framework as well. So this is advancing, although we have a poor infrastructure in the biosafety areas, I should say, because we have a limited number of people actually working in the biosafety area, which is also very important, but we should address this more properly in the future. And the two guidelines actually been developed. So this is the biosafety guidelines, and this is the national framework for Bangladesh, biosafety framework. And this is the last slide. So here is a statement from the Norman Borlaug. He said that the majority of the agriculture scientists, including myself, anticipate the great benefit from biotechnology in the coming decades to help them meet our future needs for food and fiber. So I think this is the last slide. So yeah, thank you so much. So I'm going to conclude my presentation here. So if you have any questions and other things we can discuss, so we shall go to the discussion part of my presentation. Thank you so much.
Thank you, sir. We have collected so many questions. Fewer from students and obviously fewer from the researchers, as I can say. See the questions. Sir, I need a few minutes to organize these questions,
okay.
And I will ask you. I request Shoma to do the interview section.
Shall I stop this?
Yes, sir.
sharing?
Yes, sir.
Yeah. Okay.
Stop your screen sharing, and Shoma will start the interview questions. After the interview, we go for the question answer round.
Okay.
I will ready a few questions for you.
Yeah, thank you so much.
Hello everyone, you are watching Professor Dr. Rakha Hari Sarker. He gave a very educational and informative talk on plant tissue culture, micropropagation to genetic transformation. We will now move on to the interview section of today's event. Hello sir, we are very lucky to have you with us today. We will take this opportunity to know you better. We would love to know how you built your career. Could you please share your journey as a plant scientist with our viewers?
Yeah, thank you so much, Shoma, for the question. Actually, yes, the journey is actually quite fascinating, I should say. So we start with from very elementary situation to advanced research we have been carrying out. I mentioned that I have actually completed my master's and the graduations from the Rajshahi University, where actually I was working on genetics, particularly plant genetics. And my thesis was actually on the quantitative genetics. I was working at there. So after that, at the same time, when I actually completed my M.Sc there, then I joined as a research fellow in the Rajshahi University, Department of Botany. And then actually working on the incompatibility system of brassicas. So brassica has a good breeding system, self-incompatibility mechanism. So I was actually working with our mentor, Professor Zubairi. He helped me, to understand incompatibility system, because he that time he actually returned from the US, from UK, and he was actually introduced me with the system. So I'll be working on the brassica incompatibility system. And that time actually my first paper was published in Sebrao journal. So that was my first publication in on incompatibility. Then actually in 1982 I joined Dhaka University as a lecturer. And then that time in the Dhaka University, of course, the Dhaka University Botany Department is a famous department. As you all know that, it's a big department as well. And we have a section, we have a lab, oriented research in our department. So we have, that time we have 8 to 10 labs. So our lab actually is known as the Plant Breeding Laboratory. So I was associated with the Plant Breeding Laboratory. At the same time, we come to know about the Professor Ahmed Shamsul Islam. He was actually our mentor. He's still he's 97 and he's alive. And Sir actually helped us to understand regarding tissue culture. And he in 1982 he had a big conference on tissue culture. Although that time in the Department of Botany we don't have any facilities, but he arranged the conference. And then actually the people like Thorpe and Dr. Chaturvedi from India and many famous people actually came to this conference. And so then since then actually we are actually very much interested in tissue culture. And then in 1984, you know that in Delhi it was the it was the International Genetic Congress was held. And that time actually we all participated from Dhaka, and we present our papers in the conference. And at the same time, we have been arranged a training program in NBRI, Lucknow, National Botanical Research Institute, in the lab of Professor Chaturvedi. And then actually they spent there for a month or so. Then that was actually our first training on the tissue culture. So we learned from them. Then actually we have 7 to 8 of our PhD students and younger colleagues actually there to be trained there. Then I got the Commonwealth Scholarship. I then went to England, Reading University, and my topic was actually to understand the mechanism of the self-incompatibility, because I chose that subject at that time because it was quite fascinating to me. So I chose that area to work with. And then, when I went to England, that time, this is the first time that we started the molecular biological work. Before that, I have a very little idea regarding the molecular biology. So during my PhD, we are actually trying to understand the molecular mechanism of the self-incompatibility system. Brassica has a good incompatibility system, and so people still working on incompatibility, try to understand why the plant is actually is not accepting his own pollen, for fertilization. So this is the big issue. And my supervisor was Professor Hugh Dickinson, and he's still alive, and he's emeritus professor in Oxford University. And normally, he still is my mentor. He communicated with me almost regularly. And he actually mentioned in his statement that in his profile, he said he has not been able to complete his PhD work, so he want to work the unfinished work of his PhD. Professor Dickinson actually mentioned. So I am so inspired by him, and he's a nice guy. Actually, he was younger when actually was there. He was almost 40s or so at that time, young professor, and he guided us quite a lot. And that time, my first publication was PNAS, Proceedings of the National Academy of Science, and Professor Heslop Harrison was the communicator of the article. So I'm really grateful to them, for that time that it was published in PNAS. Then after PhD, I come back to Dhaka, and then we were actually then involved in the tissue culture, because we don't have that much more facilities other than the tissue culture there that time. So also the tissue culture facilities was not that much big. We have a limited lab, and we have a very small activities there. So gradually we started the tissue culture, and particularly we started with some ornamental plants and some fast growing trees, this kind of thing, orchids as well, banana as well. So those are actually very nice material to work with. So, and also potato that time. So and Professor Islam was there, Ahmed Samsul Islam, Professor Mozammel Haq actually helped us quite a lot. And at the same time, we have some other younger colleagues like Professor Imdadul Haq, Professor Hafizur Rahman, and they are my colleagues at that time. And so they actually helped us quite a lot to develop a team. So you see, for this kind of activities, you need to have a good teamwork. We develop a team. That is very important. I should always mention that without the teamwork, without your time, you have to spend quite a lot of time to develop your own lab, your own research for these purposes. So we are lucky that time we have a senior colleagues and the younger colleagues, and also we are contemporary, the same age of us, and so we have developed the lab. And then in 90s, actually, we begin to think about transformation. So, and that time we actually associated with the ICGEB, because Bangladesh was not a member of the ICGEB, although ICGEB has been established, but unfortunately, some reason we don't know why it was that Bangladesh actually they delayed the process. So the Dhaka University Department of Botany was actually the focal point of the ICGEB in the beginning. So that was actually Professor Ahmed Samsul Islam actually developed that collaboration with the ICGEB, and Dr. Chauhan was there at that time in ICGEB, and he sent Professor Shiva Reddy to our lab for a month. So trained us regarding transformation, how to do transformation. So Shiva Reddy is actually our mentor for that. He actually trained us to develop a transformation system. Where that time we were working mainly on the grain legumes and that are also the potatoes as well. So we develop ideas from the transformation and also the molecular biology, little things that actually what are the gaps are there. So we try to procure the equipment. We try to procure our system to work with, and at the same time. After a few years afterwards, then we had another conference, an international conference, because we have an association in our department. It's the Bangladesh Association for Plant Tissue Culture and Biotechnology. With the help of this association, we develop various workshops and conferences as well. So through these conferences and workshops, we develop our skill as well, and also our ideas. Our students actually been quite benefited. Some of the colleagues that are actually working at this moment in our department actually all been, very much get benefited all these conferences from the conferences. And then at the same time, we got the money from USDA. Actually, US Department of Agriculture, that time this is the first time I received fund from USDA to work on the grain legumes for the transformation. So we started working on the lentil first, and also in the peanuts and chickpeas and mung bean. So those are the four crops actually we started. And that time, we also, develop a transformation system, we work hard to find out, how to develop a transformation system. And at the same time, we also learn the biosafety issues, during one of the conferences actually is the participants from England, from Germany, they actually give us the idea that if you want to work on the transformation, you have to have a biosafety system working in your country. Otherwise, what will you do if you develop some kind of product? What will you do in the future? So you have to go through the biosafety system, biosafety protocol as well. So you see, it's interesting that time actually Bangladesh was not even there's no idea regarding the Cartagena protocol, because this was before 2000. So Cartagena protocol actually established in 2000, and that was ratified afterwards. But before that, actually Bangladesh have an idea regarding the biosafety. So in that case, we actually get lots of help from India, from Philippines. So we have a lot of interaction with the scientists that actually come to train us. So and then we develop some kind of ideas. And in late 90s, the work is going on. Our students actually been trained. We have some PhDs, and what happened in the department. We have master's students as well. So there's quite a good team actually developed. Then I and myself and one of our colleagues, Professor Embryogenesis, went to Germany for further training on biosafety and biotechnology. That was quite helpful. And that time we actually developed collaboration with the German professors. And they used to come to Bangladesh almost every year with the collaboration, because we had some funds there at that time for USDA. So that actually helped us to bring them here and to discuss these things and to how to improve them all the time. So this is the process. And then what happened? We still got the funds for USDA for another year, another 4 years. So we got the funds from USDA for 8 years continuously. And also we develop, our own funds, and we develop lots of equipments like, minus 20, minus 80 degree freezers. Those are very important. Also PCR machines and, incubators and something like that. So this is very good, good to know that, our younger colleagues should know that, younger researchers should know how to develop a system. So this is the kind of, in our lab, we should say that is our system. We have also transformation. Why it is transformation? Because we started with the breeding, right. Then come to the tissue culture, then we come to transformation system, the understanding, and now we are doing genomics as well. So we have colleagues now in the department who are actually working on the bioinformatics and CRISPR technology as well. So recently, our colleagues have been trained from Germany. So we have been working on the CRISPR system as well, and the genetic genome editing system as well. So this is the story. So I always prefer that to have you have to develop your own skill, and you have to develop you have to spend quite a lot of time to develop yourself and to for train your students and spend a lot of time in the labs. So yeah, that's it. That's my story.
Sir, according to you, which sectors of plant science research will prosper in the near future?
Well, you see, climate change is a big issue. Mm-hmm. And particularly in our part of the world, is actually climate change is a big issue. And so I think we have to develop, some of the areas that we can address the climate changes. So particularly we design new crops. And we have to develop, genetic gain for the crops, because these crops are actually not been, familiar, they cannot address the climate issues. So we develop the, new capability of the crops. And so you have to work on this area. And also you can work on the student will be, for the future plant sciences, you have to work on the medicinal plants as well. For the development of the, in Bangladesh or India, we have lots of, medicinal plants there. We publish a journal called Plant Tissue Culture and Biotechnology. And in this journal, almost, most of the articles actually tissue culture on medicinal plants. So you see huge number of medicinal plants are there. So we can work on this area for the production of the, important products in important metabolites. and also the conservation of the plants. Tissue culture can work in this area. So tissue culture can be used for the conservation of the plants and also the development of the products. So these are the areas actually good to work. Also, due to the pandemic situation, I think we have to develop, our own, we have to design our new research areas, because the pandemic may continue for years after years. So I think our younger colleagues can think about it, how to address the pandemic issues and design their own research and something like that.
Sir, for CRISPR vector transformation, plant tissue culture techniques like protoplast culture, anther culture, cell suspension culture are regaining high demand. What are your thoughts on this?
These are protoplast culture, of course, is very good technology, but problem is that from the protoplast, development of the regeneration of the plants is still a big problem. So develop the own skill, how to regenerate plants from the protoplasts. So protoplast technology can be used for the CRISPR as well. And also, we have to develop, good transformation system for CRISPR. And also the anther culture system is very important. So these are the areas actually can be useful for, CRISPR technology to work on. And so particularly, the regeneration system is very variable. It's not consistent that you cannot have a same technology for all crops and all material. So you have to design, you have to customize your protocol according to your requirements. Right.
In the recent past, Bangladesh was in the limelight for GM crops commercialization in Asia. What is your opinion on GM crop commercialization in Bangladesh?
I have already mentioned that in 2013, Bangladesh actually permitted 4 BT brinjals. So these are actually being cultivated by farmers. I cannot tell you what is the number of the farmers, but a huge number of farmers actually being using for the cultivation. And already, the good thing is that they have, there is some economic review of this, research of this farming. So they have mentioned that they have reduced the use of the insecticide quite a lot. So they are not using, normally they use 80 to 100 times the spray the brinjals, but they reduced to a few sprays now. So this is a good opportunity that GM crops can become commercialized, and this is a good example. And also we have in Bangladesh particularly working on the lead blight resistant potatoes and also cotton, because India is a good example for cotton, GM cotton cultivation. So I think the commercialization is a big issue for the GM crops, and Bangladesh is actually working on the BT cotton at this moment. So Bangladesh will be going for cultivation of the cotton in Bangladesh as well, and we hope that the golden rice will be soon be released, soon be permitted, so the farmers can use for the golden rice. Although in the primarily what happened in the beginning, it will be not many people actually being cultivated will be cultivating this, but we can start. So there is a huge prospect, and some of the other crops like corn. Corn is in the poultry industry, the huge poultry industry, all over the places, in Bangladesh or India, in Philippines, whatever, everywhere you go, the poultry industries is developing, and also the fish industries, fish cultivation. And so these are actually, you need lots of plant material, particularly for the feeding. So for these areas, actually, we need to have commercialize the technologies, because BT corn is 80% corn is BT corn. Cotton is also more than 70% is actually BT. So these are the already commercialized, and also, soybean also commercialized. So so it will be a good thing to continue with the GM and to idea to commercialize them properly. So you need to have a good people to support. Otherwise you cannot do this. So people should understand.
So talking about GM crops, what is your opinion on the CRISPR genome editing tool, and do you consider CRISPR crops to be GM crops?
Yes, actually, recently US Department of Agriculture have announced, probably have seen that one. So they are considering CRISPR technology is not as GM. it is rather than as something else, which is actually modification, it's not following the GM technology. So they are actually approving that CRISPR as a non-GM technique. So I think it is the good to understand, science, you have to understand the science. So what happened during CRISPR, what is happening? You modified the gene of this particular plant itself. You actually edit, not introducing any gene to it. You are just editing a particular gene from the bad genes from the plant, or introducing a good gene into it. So these are actually it's not a classical GM technology. It's a different. So therefore, USDA and also USA is actually probably addressing the issue. I think hopefully in the other countries will follow them. Although maybe Europe may be a little bit stringent at this in the beginning, but they will understand the system. And of course, for Bangladesh or India, I think it is we have to consider it as a non-GM technology, and I should say that. So we can use the CRISPR for our future. Thank you.
If someone wants to start a plant tissue culture company, which plants would you consider to be useful for this purpose?
You see, plant tissue culture is always famous for the vegetatively propagated plants. Plants actually not been used for seed production, like bananas or potatoes, ornamental plants. Those are you don't need the seeds, the zygotic seeds. So what you need actually, you have to have a propagates. So these materials are actually good for the propagation, and also for the development of the clean planting material, also the disease-free planting material, like the potatoes. Potato production is huge in Bangladesh or India, or many other countries. Actually, potato production is huge, and potato demand is always there. So farmers actually need quite a lot of seed, for the cultivation. So seed tuber production through tissue culture, virus-free planting material can be developed through tissue culture. So I think it will be good to work with this, and also some of the ornamental plants they can be used for, because if you see that the huge floriculture market is developing. So there are lots of gerberas and tulips and the carnations and so on, the orchids even. So orchid is famous for tissue culture. Thailand and Singapore, Malaysia, they are all famous for the tissue culture orchid production. So these are the areas actually we can work. Orchid is good for our country as well. So you have to a little bit customize things so you can use them. So these are the areas.
In tissue culture, contamination is a problem and sometimes very frustrating. What would your advice be to the students who are facing this kind of contamination related problems?
Yes, it is. Our students, some of the students always say that all my culture has actually been contaminated. So it is really frustrating. So she or he actually developing maybe months, and then suddenly it contaminated. So you have to develop your own skill actually. What my advice is that you have to learn the techniques of handling the aseptic culture properly. So you don't go to the laminar flow and do it your own. You don't do this. So it would be good to get a good training from your senior and to understand how to handle the tools, because we use lots of tools and things like that during tissue culture, and like scissors, like the scalpels, like blades, forceps. So how to decontaminate them. So these are the ideas that you have to do this. You have to learn this, and at the same time you have to be careful choosing of the explants. So when you actually choose the explant from the outside, so you have to sterilize that properly. And also the environment, cultural environments. So if you go to the laminar flow and straight away you start with working, it's not that. So you have to open the laminar flow for quite some time, and then it will be the bench will be free from contamination. And so these are the advices. But mostly I think I will say that I would say that don't be rushed for this. For tissue culture work, you have to be very patient, and also we use fire as well. So flame. For gas flames or spirit lamp burners and things like that. So be careful that when you're actually doing this thing, you should be very careful and do it nicely and cleanly, so that the contamination, cleanliness is the priority, top priority for avoiding contamination. So these are the advice for the students.
Thank you. That was great advice, sir. You are a reviewer for many renowned journals and editor of books. Can you give us some key takeaway messages to improve manuscripts?
Good. Yes, there are a lot of preparation of the manuscript is quite a difficult task actually. So you have to follow those tasks and how to develop. Whenever actually you do a PhD or masters, you carry out a research and you want to publish it. So yes, what you have to see that what are the positive results from your thesis. So get you don't manuscript your thesis, your paper following your thesis, but rather you follow some paper as well. So you have to follow some paper. You have to particularly follow the journal, which journal you actually want to submit your article. So follow that article, and you have to go carefully. What are the positive responses? What are the positive results? And then good references as well. You have to consult the references. So what are the current references for this? So, and you have to make a good story for it, because without a story, that reviewer will not be convinced. So this is your problem, and how you have addressed it. What is your methodology, and how actually what is your outcome? So you have to have a good story out of this, and then you can publish. And also, plagiarism is also a big issue. So you have to consult that one as well. So you have to, and the other thing that. I would also, if you have problem, you can ask your supervisor. So don't prepare your manuscript without asking your supervisor. First of all, supervisor is rather experienced than you. So you should consult your supervisor, before you preparing the manuscript. And also you have to have, discussion with the co-authors. So if you have a, co-authors, so they should know that what you actually are going to write. So these are the areas actually, and also you have to follow the reviewer's advices properly. I am actually handling a journal. If I review it, or somebody actually review it, and if I give it to author, and when the author is actually giving back the journal, he or she doesn't address the properly, the issues. So whenever actually that makes the reviewer annoyed. So you have to be sure that you are actually taking the advice from the reviewer and do it carefully and have patience. Without the patience, you cannot publish it. You cannot publish it in a night actually. So these are the advice. So thank you so much.
Sir, when you recruit a research fellow, what qualities do you look for other than academic qualities?
Mm-hmm. It's a good question. Actually, many instances we actually recruit or take research fellows for our projects, and many labs actually do have these practices. And now, whenever you got the money, so we recruit the research fellows and something. Yeah. So first of all, what will be interesting to see then what is the knowledge of the particular person in the area particularly. So he or she has the knowledge of doing this. And whether he or she knows the problem. So whether what is actually material. If you have a project, and so the research fellow should know that this is the problem that I am going to work with. So first of all, these are the criteria, and also have a good skill. So if you are a research fellow, or maybe PhD fellow or something like that, so by that time you have masters or something like that in undergraduate also done. So you have somehow developed a skill. So that the skill is very important for me as well. So how we handle, in plants, we actually use lots of micro techniques. Like you go to the microscope and see cultures. You go to the tissue culture and you have to see whether the tissue culture material is responding or not. So that you have to find it out. And at the same time, skill, these are important, but also most important thing is that how the fellow is actually going to work with the other people in the lab. So you have the capability to work with the other people, because in a lab atmosphere, if you are not talking to other people, you are just on your own. This is not possible. This is not a good fellow. So we always prefer to have a good communicative attitude, and also talk to the people. And whenever you have a problem, you can talk, and then it will be good. Atmosphere in the lab, so you can adjust in your lab.
So thank you. And there is one last question which concludes our interview session. What would your words of advice be to the students and scholars who are new to the field of plant science research?
Yeah, actually, plant is a huge area. You see, if you consider from the we call microbes are also plants. You know, that some from microbiology, microbial system to plant system, and so we have a bryophyte, pteridophyte, algae, fungi, and other plants like the high plants as well. So everywhere actually you can work. So like the bryophytes and pteridophytes, people think that these are the materials, are classical materials. You don't want to work. Even the lichens, you don't want to work with the lichens. So, but these are very good. They are. I know a couple of researchers in Germany. They are actually using bryophyta for the development of the some kind of drugs. So useful drugs. So you see, all the time you can use the tissue culture technology for the bryophytes. You can use the tissue culture technology for the pteridophytes as well, because these are plants actually you cannot find them. Normally used to have them lots in the gardens or in everywhere in the forest, but they are actually being lost many ways. So I think the students should understand what is the diversity of the plant material, particularly in the plant sciences, and take each and one as a good material for their future study. And also they can use them, they hire crop materials, also they can use what type of crop is actually required for the country and so on. So these are the areas the new students, the new researchers can work.
So it was amazing experience talking to you and hearing from you. And thank you for this lovely time. And we have some questions related to today's event. It has been collected, and so let's go back to Shubho.
Okay.
So, there are a few questions I put into the chat box.
Okay. So I go into the chat box as well.
Yes, just beside the screen.
I cannot see them.
That's right. Next to the screen sharing option, there's a chat in Zoom.
Just a moment.
Take time.
Yeah. Shumi 1997 asked, sir, why callus root system working? Why is the callus system is not working why the callus system is not working in mud? What is the mud? I don't understand that. I think probably she wants to know during the root development, callus is actually produced. So for this, this question actually, you have to avoid the callus production during the root formation, because root formation is actually required a connectivity from the shoot to the root. So for this purpose, actually, you have to be careful, balancing your hormones for to avoid the callus formation during the root development. So you can avoid that one. And then pin 2 is actually, if we have more yield production food and vegetables item by tissue culture method, is there any change in the economic income or to reduce the cost of the food? Of course, if you can develop new crops with a lot of yield potentials is high, and also you can rare materials, of course you can earn more money, because also in case of the banana, we know that in case of the banana is the bunch of disease is a big disease, a devastating disease. So if you can develop the banana plant through tissue culture, you can avoid the contamination and also virus free planting material. So you can raise the economic benefit. and Dr. Saroj Kumar Mishra, Somaclon variations are targeted or random. It is rather random, I should say, because in our experience, actually, you can develop your somaclons by converting your, some of the nutrients, some of the cultures conditions. But in our experience, actually, we think that this is rather random. If you have a long time culture for your tissues, then suddenly probably you can find out then some the somaclons are developing. So it is rather a random type of system. Dr. Shobita Murthy, the secondary metabolite production is the high in callus. Is that true, sir? Have a lot of confusion. Yes, I think it is in callus. Actually, we prefer to have callus in this case because we don't want the plant as well, because the same thing will be crashing the plant and getting the somaclon. So rather, if you have a callus tissue and the callus is actually probably you can get more callus than the plant itself. So if you use the callus tissue, you can get lots of secondary metabolites. You can get them. So I think this the answer goes, and then which tissue somebody is just. I don't know. The professor, which tissue culture technique would not recommend for the Bambara groundnut legumes? Which tissue culture technology would be? Oh, it's okay. You see, the groundnut tissue culture system is maybe variable, but we are actually working on the groundnut tissue culture. We found that the normal, it's not like the normal legumes. It's a grain legumes. So you can find the lots of references in the groundnut tissue culture as well for the transformation as well. So you can use the BAP and other simple hormones like the kinetin as well for the groundnut tissue culture. You can find them in the literature. Is tissue culture appropriate for all crops? Is may not be, all crops you cannot do this, but you have to customize your crop. I don't know what is the crop actually you mentioned. You want to ask me Diriba Bayani. I don't know whatever crops you are interested. So I think we in our part of the world we are actually rice, maize. Maize is not that much, rice, and then also we are working on many other crops like brassicas and grain legumes as well, and sunflower. So these are the different kinds of crops, and of course the maize is not cultivated in Bangladesh, but maize is famous for everywhere. So you can use these are the crops, important crops for cultivation. Dr. Prabhat Kumar Singh. I am planning for the potato seed production through apical root culture, cutting using aeroponics. I think I don't have any experience on the aeroponics, but I think you can use the tissue culture potato, because we have recently visited China that they have demonstrated that they use aeroponics for the cultivation of the tomatoes. I think if the tomato is possible, I think potato is possible. In Bangladesh Agricultural Research Institute, they are actually have started aeroponics for using potato for the cultivation. I think potato could be a good material since tomato is working, so potato should be better to work with. Then we have from Dr. Mujumdar, Istiaq Hussain, can we also give storage condition, applicable condition for in vitro microtuberization, tissue culture lab? In tissue culture, actually, in potato tissue culture, we used to get a lot of microtubers. And the condition actually you have to give high amount of sucrose in the media and also maintain the light conditions. So if you do that, you can get a lot of microtubers from the potatoes. So these are microtubers actually. Normally I don't have the picture at this moment, but I can tell you that in the culture actually the tubers actually being hanging on the culture. So they are actually developing not on the media, but they are developing on the shoot as well. So you convert the shoot into a microtuber. So you can use that for tissue culture technology for the microtuber production. And then so it is possible. I am a PhD applicant, Mary Chidima Williams. Sir, I am a PhD aspirant of the plant breeding and genetics from Nigeria. Do you mind to supervise me in the PhD program in your institution? It's no problem. You can try. I don't know what is the system in your country, and also we have to see whether in Bangladesh whether it is possible or not. But I am very glad that you are interested. So you can apply for it. We used to get lots of foreign students in Bangladesh in the past, but we are not getting too much. So I think it would be a good idea that if you can apply, and we can see how we can accommodate you in our department, in our lab. Thank you so much. I think these are the questions that I have.
Yes, sir. These are the questions we can discuss over here, but there are a lot of questions I collected. There are so many questions for today's webinar, and most questions are repetitive also. So I choose those are related. Like many people are very much interested on somatic variation. Many people are interested to form a company on plant tissue culture.
Yeah.
Many people are interested on your research and your lab, and you wanted to you as a mentor. So I put one that is Williams, because I got 4 or 5 messages from this student.
Yeah, I've seen that Williams is actually interested.
Yes, sir. Many people also acknowledge you because of your slides. It's very it start from beginning to as CRISPR, or you can say the genetic modification. So it's helped them to understand the plant tissue culture. And obviously there are a lot of people, they are working on a lot of different plant, and everybody wants to know whether it is working on their plant or no. So it's always questions and query over there. We are very glad, sir, today we have you, and it's quite success. Many people at a time, 400 people or more people are watching, and I'm sure it's for YouTube it's near about 2,000 views already crossed. So it's a great a pleasure for us to have you, and you also taught few things for us that how we should behave in the lab, behave collaborate with others, because this is the angle you experience. People are looking at ours, but we don't think, we just focus on our academics, and we think that we have a marks so we can get the chance. It's also happened in India. There are a lot of students pass the NET and JRF, but they can't find a lab because it's different. Getting a fellowship is a different. There is nothing, no benefit for the supervisor. You got the fellowship for your own, but how you go to the lab is a different story. And we should focus on that way to start publication very beginning, to start focusing. And during the webinar, Soma told me that we are just know these techniques in a plant tissue culture book, but we are now watching that what happened to use these techniques. So yes, this is one type of our intention to form the bioengine.
Thing when we were in our master degree, so that's what I was saying.
Actually, when someone asks randomly all the techniques, then it's very difficult. If you know that, okay, this is the next step, this is the next step, this is the next steps, is very helpful, because generally in our BSc and MSc, the maintaining high quality lab is very difficult for 60 or 70 students working in the same lab, and there might be contaminations. So completing a full story from callus induction to regenerations is very tough in that time. And I also very happy you acknowledged few recalcitrant crops also. As I working on jute, then you know that how much phenolics and how much tough to doing the regenerations of this type of jute.
I actually forget to mention one thing that during their development, your own skill and things like that. I always mention to our student that you should love your material.
Yes.
You should feel that this is your material. You have to work with it. So if you are casual, you are not thinking that it is your own material. It is not. I said that your material will be responding when you actually think that it is yours. You love it, you do it, and handle it properly. So this is very good thing that I feel that I should mention this.
Yes, generally we also told in a casual language that these are my babies. So we generate some transgenic plants from something we call these are babies for my babies or my children. Because it lots of involvement to create these things, and it's another level of joy. Those are working on the plant tissue culture and see the regeneration from a small piece of tissue to a plant is another level of happiness, and it's totally reflect on your faces. So it's a great achievement to your success. You're doing it is for a long time, and you gather the knowledge from different sources. So it's also help us to understand that if you love the work, you can move around the world to collect the knowledge, bring into the country, and do for the betterment. And obviously we are very, very happy to progress Bangladesh in the GM crops, and hope so in future, like Philippines already granted for golden rice. I hope so in the future golden rice also available in the Bangladesh. And something happened in the Bangladesh, we Indians are very excited that we're waiting for vitivinjal in India. And so it's very, very fortunate we have you. And there are a lot of appreciation. I'm sure it's a very, very fruitful webinar we have arranged. And I once again, thank you, sir. In the future, I'm sure we meet again in this platform where try to build and try to grow slowly. So in the future, maybe we are accommodate you in another different webinar in different perspectives. But I keep in mind, I try to bring you once again in a little bit policies related talks. It's another nowadays you are lots of chairmen in lots of groups, lots of agencies. So it's another different criteria of a scientific life or experienced at the peak of your career, the policymakers. So it's also help us to understand that what in your mind about the new techniques and new things in near future. Thanks once again.
I also, thank you and also Biogen and also your efforts. We are also doing the great job. I think is this kind of seminar, particularly this pandemic condition. Actually, we are not going out. Everybody is detached. So I think this is a good way that you actually communicating the people and also listening to science at least. And also can understand what is going on in the different areas. And so I'm really grateful to you both. You are doing a good job. And so in the future, actually, I promise that from Bangladesh, actually, I will be, promoting some of our, researchers to be involved with your activities. And so in the future,
we are most welcome. Yes, we are love to hear them. We are wanted to join everyone to like 2 days. There are a lot of people from Bangladesh, Pakistan, Philippines, Nigeria, and other African, Asian countries. So it's a opportunity we have that we can learn something. And many people ask for the email address, and so share email address is available in online. So I'm not wanted to give you personally, but you can Google it, his name and University of Dhaka, you can get it, the email address.
I'm also grateful to the viewers. Actually, you mentioned so many people actually been listening to this seminar. So I am also grateful to them because in this world actually we got to understand the science particularly very precisely. Without the science, we cannot prosper our society. Because if you see, I'm talking to one. Just a moment, I will finish it. So some of the people actually do not want to have a vaccine in the beginning. So we're going to take vaccines, and so this is the science. There are some people who are actually not interested in science. So probably they don't know. They what is actually happening, and at the same time, they think that. You see, the science is how advancing, in a normal pandemic situation or in some kind of crisis, normally it takes lots of times, maybe years to develop a vaccine. But it's fortunate that it has been developed within a very short period of time. And whatever the cost is there, but we are getting the, vaccines. They're all vaccinated and gradually we will be vaccinated. So it's a huge help. So you got to understand that, science is there. So we have to promote the science. We have to, have a good avenue for the science all the time. Therefore, it will be helping us, our society. When the people are actually using insulin, they don't know how the insulin actually developed. Insulin is actually developed. It's a recombinant product. It's a GMO. So people do not know that it's a recombinant DNA product. It's a GMO, but without knowing that they are using it. So they got to understand, science. So I'm glad that, lots of young colleagues and young students and younger ones actually been attached to this seminar today. So I'm really grateful to them because I think this kind of communication we have to continue in the future. So that will help us in developing our societies. Thank you so much.
Yes. Thanks, sir. And for those participants are waiting for the password, I already pressed it and I will paste the password 1 second. We already get about 500 application for the certificate. So sir, it's a great webinar. I will send you all the rest of the questions and details about the participants and all within 1 day or 2 days for more information. So I thanks to you, and I conclude in that way that যারা বাংলাদেশ থেকে দেখলে ধন্যবাদ, আবার তোমরা ভবিষ্যতে দেখো। খুবই ভালো লাগলো সবাইকে পেয়ে, বাংলাদেশ থেকে যারা দেখেছো থ্যাংক ইউ। ইন ফিউচার তোমরা দেখতে থাকো, আমরা চেষ্টা করব আরো এরকম ওয়েবিনার আনতে, যাতে আমরা সবাই মিলে শিখতে পারি। একটা প্ল্যাটফর্ম তৈরি হতে পারে পৃথিবীতে, যেটা ওপেন হবে সবারই জন্য আর ফ্রি হবে সবারই জন্য। থ্যাংক ইউ। Thanks.
So Bangladesh is particularly hospitable. So I'm inviting you, both Shubhabrato and Soma, to Bangladesh whenever it is possible.
Yes, sir.
Please come to our lab even.
Yes,
so they will be love to have you in our lab. So
definitely.
I will just tag on to him.
শেষের কথাটা শোনা,
মানে যেই শুভ কোনো অপরচুনিটি পায় বাংলাদেশ যাওয়ার, আমি একদম ওকে আর ছাড়বো না, ওর সঙ্গেই চলে যাবো।
Definitely, you are welcome.
হ্যাঁ, আর আমি জুটে কাজ করি, ভবিষ্যতে করার ইচ্ছা আছে, তো জুট তো বাংলাদেশেও আছে, তো মেবি কখনো তো যেতে।
Yeah, of course, jute a lot of activities is going on as well in development.
Yes, তো দেখা হবে, গেলে তো অবশ্যই দেখা হবে। আপনি এলেও ওয়েলকাম, আমরা কলকাতাতে।
I used to visit Baligans,
আমার ল্যাবটা তো বালিগঞ্জে, আমাদের ল্যাব।
Yeah, whenever possible I will go.
Yes.
And give my compliments to Sir Thapan sir.
Yes.
Because I know that he is our mentor. He also visited Bangladesh so many times, and in many conferences and meetings, he is the top selection for us. So you are, and recently he has a post. I think one of our senior journalists actually quoted him regarding golden rice issues and things like that. Everyone knows Shravan Datto very well.
Yes, and his work in golden rice is very much motivating for us also.
Okay.
Thank you, sir. Thanks for
All right. Okay.
Namaskar.
All
Sir.
right.
Bhalu
Namaskar.
thakben.
Yeah.
Thanks all the participants who have joined for today's webinar. We are waiting for more webinar in the future. Our next webinar is on 16 August on the wheat breeding. We also are eminent scientist, retired professor. So we are hoping you all guys are catch up in that webinar. Thanks for the today's event, make it so successful. Thanks everybody.
The next webinar is on 16th, and please note the time. It is in the evening. Our webinars are generally in the morning, Indian Standard Time, but this one is it is at 7 p.m. Please note the time.
And sorry, those are don't offend it. There are a lot of questions, and I'm sure during the question answer round and the interview, you got the essence of your answer. If you not get it, I will try to bring the answer in the different way. And obviously, I forward your question to our speakers. Thanks. And this event is end now. We're going to offline. Please submit your feedback as soon as possible, at least within 5 hours from now. Thanks. See you in our next webinar. Those are waiting for now and just.
It's recording now. I mean, loading on your laptop. Right?
No. I'm still waiting.
Your Zoom is on.
Yes, no problem.
And I would suggest that you just bring your laptop here and do the rest of the work from here.